Welcome back to The Deep Dive, the show that takes foundational, highly detailed source material, the core knowledge you need, and distills it into the essential truth, giving you the complete, accelerated picture.
Today we are tackling, really, the very starting line of life science.
That's right, the fundamentals of cell and molecular biology, drawn directly from Chapter 1 of Karp's foundational text.
And our mission here is to give you a fast-paced but also highly detailed understanding of what cells are, how they work, and their complex evolutionary journey.
This is such a critical deep dive. I think cell biology can feel daunting for a lot of people because it deals with complexity on a scale that's just hard to wrap your head around.
It is, but our goal is to show you that mastering this material, every property, every mechanism, every experiment, it provides the necessary conceptual framework for, well, all of biology that follows.
So you can grasp the complexity without, hopefully, the information overload.
Exactly.
Okay, let's unpack this immediately. I think the best place to start is with a really powerful visualization.
Yes. I want you to picture these remarkable, incredibly detailed, hand-drawn images of nerve cells.
And these weren't made by a computer, right? This is old school.
Not at all. They were created by the Spanish histologist Santiago Ramon y Cajal in the late 19th century. He was looking at slices of a cat brain, and his drawings showed these individual branched structures. I mean, discrete cells with very definite boundaries.
And that was revolutionary at the time.
Completely. This was a real scientific battleground.
Because Cajal's work was establishing the neuron as an individual cell, and that directly challenged the prevailing theory from his rival, Camillo Golgi.
Right. Golgi, who insisted the brain was this single, continuous, sort of connected network.
This was a major fight.
It was. And Cajal's victory, establishing the neuron as a discrete entity. It highlights the central theme of this entire chapter.
Which is?
The cell is the fundamental discrete unit of life. It's not just about structure. It's the boundary between self and non-self.
Precisely. And as the chapter title suggests, we are cells. They're the core of our existence. They dictate how we grow, how we age, and, how we experience disease.
Absolutely.
So today, we're going to move quickly but thoroughly through the timeline of discovery, detail the 9 essential properties all cells share, and then look at that fundamental evolutionary split into 2 major classes.
We'll also examine the peculiar status of viruses, see how multicellularity first arrays with something like Volvox, and then finish with the cutting-edge concepts in tissue engineering and stem cell therapy.
Let's start at the beginning then. Section 1.1, the discovery of cells.
We have to begin with the immense conceptual challenge of scale.
Right.
Cells and the structures inside them are just far too small for our naked senses. You can't see them, you can't hear them, you can't touch them directly.
So the whole field is really a testament to human ingenuity.
It's an immense tribute to human creative intelligence. The cleverness required to devise instruments and techniques to study these worlds within worlds.
The source draws a really wonderful contrast here. It puts cell biology, in terms of what it takes to study it, on the same level as astronomy.
Or nuclear physics.
Exactly. Building massive telescopes to see primordial galaxies 13 billion light years away, or giant colliders to find the Higgs boson.
It's the same creative leap. We're using equally elaborate techniques to study the smallest components of life and the largest components of the universe.
In this creative study, it leads to the field's defining philosophy, which is that cell and molecular biology is fundamentally reductionist.
Yes, reductionist. The underlying assumption is that we can explain the character of the whole living organism, its behavior, its complexity, by understanding its parts.
The mechanisms, the machinery.
Precisely. And while that might, take away some of the initial wonder in history, it replaces it with a far deeper appreciation for the sheer complexity and elegant beauty of it all.
Of course, this reductionist journey needed technology. We had curved glass for spectacles way back in the 13th century, but the compound microscope really only emerged toward the end of the 16th century.
And the actual discovery of cells, that's credited to Robert Hooke in 1665.
Hooke was the curator of the Royal Society of London, and he was looking at thin slices of cork bark. He was trying to figure out why cork is such a good bottle stopper.
Right. And figure 1.1a in the text gives you this fascinating look at his ornate compound microscope and his famous drawing.
What he saw was this honeycomb-like network of repeating pores.
And he called them cells because they reminded him of the small, simple rooms, the cellae, that monks lived in.
But it's so important to remember he wasn't looking at living tissue.
Not at all. He was observing the empty, rigid cell walls of dead plant material. structures that were built by living cells before they died.
The story then shifts pretty dramatically with Antonie van Leeuwenhoek.
Ah, yes, the Dutch amateur microscopist. He sold clothes and buttons for a living, but his hobby was grinding these incredible single-lens microscopes.
And his were actually better than Hooke's, right?
Oh, far superior. Figure 1.1b shows one. They could achieve 270 times magnification and a resolution sharp enough to see things about 1.35 micrometers apart.
And with that kind of precision,
He became the first person in history to see living cells.
he looked at everything. Pond water teeming with what he called animalcules, sperm cells.
He even scraped bacteria from his own teeth and looked at them. The Royal Society was pretty skeptical at first, getting these reports from an amateur tradesman in Holland.
But they sent Hooke to check it out.
They did. And when Hooke confirmed his observations, Leeuwenhoek became a scientific celebrity.
So these foundational observations, they paved the way for what we now call the cell theory.
But it took a while. It wasn't until the 1830s that scientists really synthesized these findings and understood the immense importance of the cell.
You have Matthias Schleiden, a botanist, who concludes in 1838 that all plants are made of cells and that the plant embryo arises from a single cell.
And then, in 1839, his colleague Theodor Schwann, a zoologist, extends that principle to the entire animal kingdom.
And he proposes the first two fundamental tenets that we still use today.
Tenet 1. All organisms are composed of one or more cells.
And tenet 2. The cell is the basic structural unit of life.
But the theory wasn't complete. And here's where a really critical historical correction had to be made.
Right, because Schleiden and Schwann still believed that cells could just appear from non-cellular materials.
It was like the old idea of spontaneous generation. They were still holding on to that.
Until Rudolf Virchow.
Exactly. A pathologist who, in 1855, made the convincing argument for the third crucial tenet.
Which is?
Cells can arise only by division from a pre-existing cell. The famous Latin phrase, omnis cellula e cellula.
And now, with modern molecular biology, we've sort of naturally added a fourth tenet.
Right, one that deals with the information.
That cells contain genetic information in the form of DNA, and that critical information is passed faithfully from the parent cell to the daughter cell.
And that sequence discovery, formulation, correction, that's really the true foundation of the field.
So that foundation brings us to section 1.2, the basic properties of cells. If cells are the structural unit, they're also the smallest unit to exhibit life itself.
We know this because you can take isolated whole cells from a body and culture them in vitro. They can grow and reproduce completely outside the body.
And on the flip side of that, death is also a basic property of life.
It is.
Yeah.
In a multicellular organism, cells often die not from random damage, but through a highly regulated, programmed internal mechanism.
Apoptosis.
Apoptosis. It's the cell's way of eliminating itself when it's no longer needed, or critically if it poses a cancer risk.
And if we're going to talk about cell culture, we have to talk about the legendary HeLa cells.
Absolutely. These are the first human cells ever successfully cultured in vitro. This was back in 1951 by George and Martha Gay.
And they were derived from a malignant tumor from a woman named Henrietta Lacks.
Yes, and figure 1.2 shows these cells, which are still being cultured today by the trillions all over the world. They are, for all intents and purposes, immortal in culture.
Unlike normal cells, which have a finite lifespan.
A very finite lifespan. This robustness makes them an absolutely essential tool for cell biologists. They're readily available and infinitely reproducible.
Okay, let's dive into the first and maybe the most striking property. Property 1. Cells are highly complex and organized.
When we say complexity here, we mean order, consistency, a minimal tolerance for error, and extensive internal regulation.
The precision is just staggering.
It is. Think about this. When a cell duplicates its DNA, the initial error rate is less than one mistake for every 10 million nucleotides.
And even those few mistakes are usually caught and fixed.
By elaborate repair mechanisms. The machinery isn't just complex, it's incredibly precise and resilient.
And this complexity exists across these huge scales. Figure 1.3 illustrates this hierarchy so well using an intestinal epithelial cell.
It does. You can visualize the layers. You start at the tissue level, looking at a stained villus from the small intestine.
Then you zoom into inset 1, and you see the epithelial cell layer itself, where individual cells are stitched together tightly, forming a barrier.
Inset 2 shows the surface of those cells, covered in these finger-like projections called microvilli.
Which just dramatically increase the surface area for absorption.
Then you look inside those microvilli, inset 3, and you see Their internal skeleton is made of these ordered bundles of actin filaments.
And those filaments are themselves polymers of protein monomers.
Then you look deeper into the main body of the cell, and you find huge numbers of mitochondria, the powerhouses, in inset 4.
And even inside the mitochondria in set 5, you see these stalked particles coming off the inner membrane.
And then the final zoom in set 6 and 7, reveals those particles as the highly organized ATP synthesizing machinery, a literal molecular turbine generating this cell's energy currency.
So you go from a whole tissue all the way down to a single molecular machine. It's an incredible hierarchy of organization.
What's really astonishing is the evolutionary consistency. That apin and those ATP synthesizing enzymes are virtually identical, whether you look in a human, a snail, yeast, or a redwood tree.
So these basic processes are conserved across all life.
Which is exactly why we can use such a diverse array of model organisms to study cell biology.
Okay, this is where it gets really counterintuitive for me. The idea of stochastic events and biased randomness.
Right.
We use words like machinery, which implies this clockwork precision, but the reality is that molecules inside the cell are just moving randomly because of thermal energy.
And yet the cell somehow harnesses that random movement for highly directed, purposeful outcomes.
This conflict is perfectly captured by Levinthal's paradox from 1969.
Yes, so proteins are built as these long chains of amino acids, and to be functional, they have to fold into one very specific 3D structure.
And Levinthal calculated that if this folding was just a random search of all possible shapes,
It would take longer than the age of the universe to find the right one.
it's the ultimate impossible problem. The analogy they use is a monkey at a piano randomly hitting keys and expecting it to compose a Beethoven sonata.
It's just not going to happen.
So what's the resolution?
The resolution, which tells you so much about cellular organization, is that folding is not purely random. It's a stepwise process.
Okay.
It follows specific pathways. The formation of one partially folded intermediate guides the chain rapidly towards the next, more structured intermediate.
So the random thermal activity is biased.
Exactly. It's directed along specific energy pathways, which allows the correct structure to be found in milliseconds. It's a fundamental organizational strategy.
Okay, property 2. Cells possess a genetic program.
This information, encoded in DNA genes, is the blueprint. It dictates the construction of structures, gives directions for activity, and it's the program for self-replication.
The scale is hard to grasp. If you wrote out the human genetic program, it would fill millions of pages of text.
And all of that is packaged into chromosomes inside the tiny nucleus. And, importantly, the structure of DNA allows for mutations, which create the variation that drives evolution.
Property 3. Cells are capable of producing more of themselves.
This happens through division. It requires faithful duplication of the genetic material, followed by this careful distribution of that material and all the other cellular contents into two daughter cells.
But division isn't always equal, is it?
No, and that's a key point. Think about the human oocyte, the egg cell, shown in Frag 1.4. Its division is highly unequal.
It keeps almost all the cytoplasm.
Exactly. All the resources needed for early development are kept in the large oocyte, while the other product is just this tiny, non-functional polar body.
But the genetic material is split evenly?
The genetic material is always split equally and completely.
Okay. Property 4. Cells acquire and utilize energy. Every single biological process needs energy, and almost all of that energy starts with the sun.
Right. For photosynthetic organisms, like plants and algae, you have photosynthesis. Figure 1.5 shows the chloroplast and the alga spirogyra. Light energy is trapped and converted into chemical energy, stored in carbohydrates like starch.
And for animals like us, the energy arrives prepackaged as, say, glucose.
And that glucose is then disassembled inside the cell, and the energy released is stored in this readily available universal energy currency molecule,
ATP.
ATP, which is then used to fuel everything else.
The book also mentions this massive amount of turnover.
Yes, cells spend enormous energy constantly breaking down and rebuilding their own parts. This isn't wasteful. It's essential for maintaining integrity, and crucially, it allows the cell to respond very quickly to changes in its environment.
So property 5 and 6 combine action and machinery. Cells carry out a variety of chemical reactions and engage in mechanical activities.
Think of a cell as the most sophisticated chemical factory you can imagine. Even a simple bacterium performs hundreds of different chemical transformations.
Using enzymes.
Using enzymes, highly specific catalysts, that speed up reactions by factors of billions. The sum of all these reactions is the cell's metabolism.
And that metabolism powers constant mechanical activity.
Things like internal transport, the assembly and disassembly of structures, and cell movement. These all rely on motor proteins.
Molecular machines that literally change shape and walk along cytoskeletal tracks to move things around.
Exactly.
Properties 7 and 8 deal with communication and control, response to stimuli, and self-regulation.
All cells have these highly specific receptors on their surfaces and inside that interact with external signals, hormones, growth factors, signals from neighbors.
And the response could be anything from changing its metabolism to moving or even initiating apoptosis.
And this external communication is key to the cell's internal robustness, its ability to maintain order.
Right. Cells are durable because they're constantly self-regulating.
They use these intricate feedback circuits that automatically detect and correct fluctuations. When that regulation breaks down, if a cell ignores signals to stop growing, for instance,
That's when you get cancer.
that's the pathology we call cancer.
The classic illustration of this self-regulation is Driesch's sea urchin experiment from 1891.
It's a fantastic example. Driesch was working with early sea urchin embryos, and when he deliberately separated the first 2 or 4 cells of an embryo,
They didn't form a quarter or half of an embryo.
no, each isolated cell regulated its destiny and formed a normal, complete, just smaller embryo.
That is profound. The isolated cell somehow recognized the absence of its neighbors and completely redirected its developmental program.
It's an incredible demonstration of self-regulation.
Finally, property 9, cells evolve.
While the origin of the very first cell from non-living material is still one of biology's great mysteries, the evolution after that is clear.
All living organisms share fundamental common features: the same genetic code, a plasma membrane, ribosomes.
Which means they all evolved from a single common ancestral cell that lived more than 3 billion years ago.
The last universal common ancestor, or LUCA.
And evolution is not just ancient history. It's happening right now, as we can see with the growing crisis of antibiotic resistance in bacteria.
That discussion of LUCA brings us directly to the major evolutionary split that defines life on Earth. Section 1.3. 2 fundamentally different classes of cells.
Right. Once the electron microscope revealed the internal structures, this basic dichotomy became obvious. You have prokaryotic cells,
Which are simpler and include bacteria and archaea.
and then you have eukaryotic cells, which are much more complex and include protists, fungi, plants, and animals.
The timeline in Figure 1.9 shows just how early this split happened. Prokaryotes go back at least 2.7 billion years.
And complex animals only appear relatively recently, about 600 million years ago.
Despite the vast structural differences, which are summarized in Table 1.1, the similarities are actually pretty profound.
They are, and they reflect that common ancestry from LUCA. Both cell types share the same genetic language, the DNA code. They use similar mechanisms for transcription and translation. They share core metabolic pathways.
Like glycolysis in the TCA cycle.
Right. They also have similar plasma membrane construction and even possess similar cytoskeletal filaments made of actin-like and tubulin-like proteins.
But the differences are where the complexity really diverges. Eukaryotes have a true nucleus.
A specialized compartment for the genetic material, bounded by a double membrane called the nuclear envelope, which is perforated with these complex pores that regulate traffic.
Whereas prokaryotes, the name says it all. Pro meaning before and karyon meaning nucleus. They just have a nucleoid.
A poorly demarcated region with no surrounding membrane.
Eukaryotic DNA is also organized differently. It's complex, linear, and tightly associated with proteins to form chromatin.
While prokaryotes typically have a single circular chromosome.
And then there are the organelles. The eukaryotic cytoplasm, shown in Figure 1.10, is this bustling city filled with membrane-bound compartments, mitochondria, the ER, Golgi lysosomes.
This compartmentalization is key. It allows specialized activities to happen at the same time without interfering with each other. Prokaryotes are largely devoid of this internal membrane system.
And what about cell division?
Radically different. Eukaryotes use the complex, precise process of mitosis, with an elaborate mitotic spindle made of microtubules to segregate their chromosomes.
Prokaryotic division is much simpler.
Much simpler. And reproduction is different too. Eukaryotes are typically diploid and undergo sexual reproduction, while prokaryotes are haploid and non-sexual, though some can do conjugation.
Okay, let's break down the prokaryotes. There are 2 domains, archaea and bacteria.
Yes, and phylogenetic analysis actually places archaea evolutionarily closer to us, to eukaryotes, than they are to bacteria.
Archaea are famous for the extremophiles, right? organisms that live in boiling water or super salty lakes.
Exactly. Hyperthermophiles, halophiles, acidophiles. But it's important to remember many archaea live in normal environments too.
The other domain, bacteria, includes everything from the tiniest cells, mycoplasma, to the most complex prokaryotes, the cyanobacteria.
Cyanobacteria are critical. They have these elaborate internal membranes for photosynthesis, seen in figure 1.15a, and they're the ones who originally oxygenated our atmosphere.
They're also pioneer organisms capable of nitrogen fixation.
This extreme diversity leads to a really important sidebar: prokaryotic diversity and the human microbiome.
Right, because traditional lab methods only identified a tiny fraction of the species that actually exist.
Less than 0.1%, it's estimated. But modern molecular techniques, like sequencing the 16S RNA gene, let us study diversity without needing to culture them.
This led to the concept of the metagenome.
Right, sequencing basically all the genes in a given habitat, like the human gut. The results are staggering.
We carry trillions of microbes, and the collective genes in our gut microbes outnumber our own human genome by about 150 times.
And their functions are vital. Vitamin synthesis, breaking down complex sugars, preventing pathogens from colonizing, they're essential partners.
Okay, moving back to eukaryotic cells. The most structurally complex eukaryotes are often not in multicellular organisms.
No, they're often the single-celled protists, like Vorticella in figure 1.1. All the complex machinery for sensing, feeding, and moving is packed into that one cell.
The other evolutionary path, of course, led to multicellular organisms through differentiation. A fertilized human egg gives rise to about 250 distinct cell types.
And this brings us directly to a key clinical section: the human perspective on cell replacement therapy.
The goal here is simple in concept. Many diseases, like Parkinson's or type 1 diabetes, are caused by the death of specific cell types. The idea is to replace them.
And the most established version of this today is bone marrow transplantation.
Used for blood cancers. The patient's own blood-forming cells are destroyed and then replaced by donor hematopoietic stem cells, or HSCs.
These HSCs can reconstitute the entire blood system.
This brings us to modern therapies, starting with adult stem cells, or ASCs.
These are undifferentiated and self-renewing, but they are multipotent.
Meaning they can only differentiate into a few related cell types.
Right. A great example is the clinical trials with cardiac stem cells. Infusing them into patients with heart damage led to significant replacement of damaged muscle.
And the huge advantage of ASCs is that they are autologous, taken from and returned to the same patient, so no immune rejection.
Exactly. The next class is embryonic stem cells, or ES cells.
These are pluripotent, meaning they can differentiate into every cell type in the body.
They hold the greatest potential, but also the greatest challenges.
There are major ethical concerns about their source, plus technical risks like forming tumors called teratomas and, of course, immune rejection.
Which brings us to the conceptual breakthrough. Therapy number 3, induced pluripotent stem cells, or iPS cells.
This was Shinya Yamanaka's discovery in 2006. He showed you could reprogram a fully differentiated cell, like a skin cell, backward into a pluripotent state.
Just by introducing 4 key genes, OCT4, SOX2, KLF4, and MyThec, this provides an unlimited supply of patient-specific cells without the ethical issues of using embryos.
iPS cells are now crucial for disease modeling, creating patients in a petri dish.
But they're not perfect yet. They sometimes show genomic abnormalities and retain some memory of their original cell type.
And finally, the most recent development is direct cell reprogramming, or transdifferentiation.
This is even more efficient. It converts one differentiated cell type directly into another, like a pancreatic acinar cell, into an insulin-producing beta cell without passing through that risky pluripotent state.
It raises the prospect of replacing disease cells right inside the body.
Now, to manage all this research, cell biologists rely heavily on model organisms.
Because the basic processes are so conserved across species, figure 1.8 shows the 6 key ones.
You have the bacterium E. coli, the yeast Saccharomyces cerevisiae, the plant Arabidopsis thaliana, the nematode C. elegans, the fruit fly Drosophila melanogaster, and the mouse musculus.
Each one is a specialist tool for answering different biological questions.
And we should briefly touch on cell size and constraints. Why are most cells microscopic?
There are 3 main reasons. First, gene limitation. One nucleus can only produce so much mRNA.
Second, the critical surface area to volume ratio. As a cell gets bigger, its volume increases much faster than its surface area.
So it can't exchange enough nutrients and waste to support its own metabolism.
And third, diffusion time. Diffusion is fast over short distances, but becomes incredibly slow over longer ones. A large cell just couldn't transport materials efficiently internally.
This idea of the cell as a machine has given rise to a new field, synthetic biology.
The goal here is to use cellular parts as building blocks to create minimal living cells or entirely new life forms.
J. Craig Venter's team famously did this in 2010, replacing one bacterial genome with a chemically synthesized one, effectively changing one species into another.
And this has immediate applications, like engineering bacteria to produce biofuels.
So this concept of a minimal life form takes us to our next section, viruses and viroids.
Right. Before the late 19th century, everyone thought all infectious diseases were caused by bacteria.
That changed in 1892 when Dmitry Ivanovsky showed that sap from diseased tobacco plants was still infectious even after being filtered to remove bacteria.
He concluded the pathogens were smaller and simpler viruses.
And in 1935, Wendell Stanley crystallized the tobacco mosaic virus, TMV, proving it could be an inactive chemical but still infectious.
And we now define a virus as an obligatory intracellular parasite.
They cannot reproduce or metabolize outside of a host cell.
Outside the cell, they exist as an inanimate particle called a virion. It's basically a macromolecular package,
Yeah.
a simple structure, genetic material, either DNA or RNA, surrounded by a protein shell called a capsid.
And they achieve this amazing economy of genetic information by building that capsid from many identical copies of just one or a few proteins.
The diversity is huge. You have icosahedral shapes like adenovirus, some with a lipid envelope like HIV, and then the incredibly complex bacteriophages that infect bacteria.
And they're very specific about their hosts, aren't they?
Very. It's determined by surface proteins on the virus that have to bind to specific receptors on the host cell.
A great, if terrifying, example of this specificity adapting is the 1918 influenza pandemic.
It killed over 30 million people. Researchers were able to reconstruct the virus from preserved tissues of victims buried in Alaskan permafrost.
And the analysis showed it jumped from birds to humans, adapting with just a few key mutations that made it incredibly virulent.
Viral infection follows two main paths. First is the lytic infection.
Where the virus hijacks the host cell, makes copies of itself, and then ruptures or lyses the cell to release the new viruses.
The second type is integration, where the viral DNA inserts itself into the host chromosome, becoming a provirus.
This can be dormant, or in animal cells, it can turn the cell into a continuous virus factory. And sometimes this integration leads to cancer.
And if viruses are masters of reductionism, viroids take it even further.
Discovered in 1971, a viroid is a small circular RNA molecule that has no protein coat at all. It's just naked infectious RNA.
And it doesn't even encode any proteins. It just hijacks the host cell's own machinery to get itself copied, causing disease by interfering with the host's gene expression.
The simplicity of prokaryotes raises that huge evolutionary question. If they came first, how did they give rise to the complexity of eukaryotes?
And the answer, for the most part, is the Endosymbiont Theory.
Championed by Lynn Margulis, it posits that mitochondria and chloroplasts evolved from smaller prokaryotic cells that took up residence inside a larger host cell.
The evolutionary stages seem clear. You start with an anaerobic prokaryote. It develops internal membranes, creating a nucleus. This is the first eukaryotic common ancestor, or FECA.
Then stage 3 is the big one, the acquisition of mitochondria.
A descendant of the FECA ingests an aerobic prokaryote, which resists digestion and becomes a permanent endosymbiont. This happened early because almost all eukaryotes have mitochondria.
Then you get the last eukaryotic common ancestor, or Lessiae, which develops all the other shared eukaryotic traits.
And finally, a descendant of LEC ingests a cyanobacterium, which evolves into the chloroplast. This happened later, only in the plant and algal lineage.
How do we even know this?
By comparing nucleotide sequences. In the 1970s, Carl Woese compared ribosomal RNA sequences.
And what did he find?
He found that the chloroplast's RNA sequence was much closer to a cyanobacterium's rRNA than it was to the host cell's nuclear RNA. It was a slam dunk piece of evidence for endosymbiosis.
And his work also completely redrew the tree of life.
It did. It established the three domains, Archaea, Bacteria, and Eucarya. And it showed that Archaea are actually closer to us, the Eucarya, than they are to bacteria.
But modern sequencing has complicated that beautiful, clean tree.
With the discovery of horizontal gene transfer, or HGT, we found that genomes were mosaics. Genes were being transferred horizontally between species, not just inherited vertically.
This led to the complexity hypothesis.
The idea that informational genes, like RNA, are hard to transfer laterally, but metabolic genes are much easier.
And that's what the data shows.
It does. It means the eukaryotic genome is a true mosaic. The informational core seems to come from an archaeal ancestor. But the metabolic genes have a strong eubacterial character. No single, simple tree can tell the whole story.
That historical complexity is a nice contrast with the more recent, independent evolution of multicellularity.
Which arose multiple times in different lineages.
Volvox is the classic textbook example of this.
It is a gelatinous sphere of thousands of cells. But what makes it truly multicellular and not just a colony?
It has to meet two criteria. First, it exists and propagates only as a group. And second, there's true differentiation.
Different cells take on specialized functions. In Volvox, most cells handle movement and photosynthesis. But a select few, the gonidia, are set aside solely for reproduction.
And they have that bizarre developmental process where the embryo has to turn itself inside out.
The inversion, yes. To get its flagella facing outward so it can move. It's a fantastic system for studying how multicellularity evolves.
Finally, let's explore the practical application of the cell as a building block idea in tissue engineering.
The goal here is to use living cells to build functional organs and tissues, increasing biocompatibility compared to, say, an artificial kidney.
The core strategy is basically advanced cell culture. You grow cells on a 3D pattern substrate called a scaffold.
Exactly. Figure 1.25 shows a replacement blood vessel grown this way.
The first challenge is getting the right cells. Ideally, autologous cells from the patient to avoid immune rejection.
Which is where iPSC technology is becoming so powerful, you can induce the precise cell type you need.
The second challenge is the scaffold itself. It needs to be biodegradable so the patient's own tissue eventually replaces it completely.
And, most critically, the scaffold must be porous.
Why is porosity so vital?
2 reasons. It provides much more surface area for cells to attach to. But more importantly, it ensures that cells deep inside the structure can get the nutrients and oxygen they need, overcoming that diffusion limit we talked about.
So people are using techniques like electrospinning to create fiber meshes, or even 3D bioprinting to print cells and matrix material at the same time.
It's still largely in the research stage, but it holds immense promise for the future.
This has been an incredibly detailed and, I think, essential deep dive into the fundamentals of cell and molecular biology.
I agree. We've navigated the history, from Hooke and Leeuwenhoek to the establishment and correction of the cell theory.
We meticulously detailed the 9 properties of cells, focusing on their remarkable organization, the resolution of Levinthal's paradox, and that sophisticated self-regulation shown by Driesch's experiment.
We tackled that fundamental evolutionary split into prokaryotes and eukaryotes, highlighting the sheer scale of the global microbiome.
And we connected it all to clinical reality, exploring the generations of stem cell therapy, from HSCs to the revolution of iPS cells, and how that knowledge is now being applied in tissue engineering.
We established that the eukaryotic genome is a mosaic, shaped by vertical inheritance, endosymbiosis, and horizontal gene transfer.
So here's the final thought for you to carry forward, building on that concept of the evolutionary composite.
We learned that viruses exist as inanimate, non-metabolizing packages, yet they're made of the same molecules as our living cells. And we know that nearly all existing eukaryotes are descendants of a cell that incorporated a mitochondrion.
So this raises an important question.
Considering the pervasive influence of horizontal gene transfer and ancient symbiosis, how much of your current cell function is purely inherited vertically from LUCA? And how much is comprised of functional genetic material acquired through those ancient symbiotic or lateral transfers?
How much of the complex, highly regulated machine that is you is an evolutionary composite built and maintained by borrowed parts?
A fascinating and complex thought to leave you with.
Thank you for joining us for the deep dive.
We appreciate you trusting us with your learning.
And a warm thank you from the Last Minute Lecture team.